December. Okay, let's begin with these three terms. I briefly introduced them already. Do you now all, if you had an examen and these terms flashed by you, would you feel ready to explain what the difference is? I have a method that uses homology, that uses de novo, or that uses ab initio. Can you sort of associate something with it, or should I go through it again? Who says go through it again? Okay. You're the only one who at least partially sort of gave an answer. So again, by homology, if I want to infer some feature such as protein function by homology, then ultimately I use this idea that evolution selects proteins to work similarly. Similar proteins work similarly. There's an evolutionary constraint to do that. That allows me to look at sequence similarity and infer from sequence similarity in function. That's by homology. That means that for one of these two, for which I have similar sequences, I need to know something about the function, right? De novo means that I'm going to try to, for instance, train a machine learning device. In the machine learning device, I have thousands of proteins for which I know, for instance, their subcellular localization. And I hope the machine learning device extracts some generic rules from these thousands of proteins. And these generic rules are such that if I came with a new protein that is not similar to anything for which I have an experimental annotation, machine learning can predict subcellular localization. That's de novo. Ab initio really is the idea that you have some principles, and based on these principles, you can predict. This is the Newton's equations, and with Newton's equations, essentially, you can predict the movement between the sun and Earth, for instance, or the sun and the planets. Now, in biology, we have very few of these examples, if any. So the goal is to predict subcellular localization. Subcellular localization gets us back into when you open a cell, if you could slice through a cell, this is a drawing from David Kutzel, an illustration. What you would see, first of all, this green thing here, the cell is surrounded by a membrane, by a lipid layer or a double layer of lipids outside that protect the cell and sort of distinguish between inside and outside. Secondly, what you will see is that there is a high density in the cell. There's a lot of substructure. So this particular case here is mycoplasma, and the substructure in that sense is not organized substructure. We get to that in a minute. But essentially, you see, for instance, these fibers, tracks here, on which, in fact, material is moved through the cell. There's a bacterial cell, highly densely packed. Now, in bacterial cells, essentially, as I said, as I indicated, you do have a substructure, so you have these tracks on which things are moving. You have some plasmids, you have ribosomes, but they all sort of are assumed to somehow swim in this thing. So this whole cell is like a bag, and in this bag, these substructures swim around. Now swim, again, the density is high, is a solid state, it's not water, it's not swimming around like you would in water, but it is moving around. And this, again, bacterial cells have membranes already. Some of them have the flagellum that allows them to move, to orient themselves toward more nourishing food source, for instance. And some bacteria have two levels of outer membranes. Now, in contrast, the eukaryote, karyot is the Greek word for core. Euka is with a core, so that's a procar. That's before cores were invented. And the core is what we in Latin say the nucleus, or in English it's also referred to as the nucleus. So this nucleus, can anybody remember? I briefly discussed what is special, what is done in the nucleus. Can anybody remember that? So in the nucleus, essentially, first of all, you have a subcompartmentalization. So meaning this nucleus in itself has a membrane now going around, in fact even two. Okay? So inside the cell you have sort of another cell if you want. This is the overstatement a little bit. Let's go here to the blue ones here. These are mitochondria. Mitochondria create energy. Now, mitochondria are assumed to be sort of taken up from bacteria. So they essentially are bacteria that are swallowed by the eukaryotes, are enslaved to do the energy production for a eukaryote. That's the way we see them. There is a mitochondrial genome, but that codes for just a bunch of genes, a couple of them. There are about 2,000 proteins working in the mitochondria, but they actually come from the genome that is the core genome of the eukaryote. The core genome is in the nucleus. So why do you believe it has an advantage to introduce in evolution such an organization? Why? So in the bacteria have an immense level of complexity, and they're very, very successful, arguably, in evolution. So why not continue this concept? Why go onto this level of a subclassification? Why introduce another cell in a cell, so to speak? The nucleus, for instance. What could be the advantage of that? Any idea? Yeah?
Maybe it's an additional layer of protection because the genetic material is very important.
That's one answer. So it's an additional layer of protection. So any intruder will not immediately get at the DNA and will not get into the machinery. So it's not only that the DNA is there and is protected. Possibly, more importantly, the entire machinery of reproducing the DNA, duplicating the DNA, using the DNA to build proteins is protected. So what typically those viruses or bacteria that intrude into eukaryotes do is they inject their DNA. And they use the eukaryotic cell to make the eukaryotic cell produce their proteins. So typically viruses don't have enough genetic material to produce proteins. They enslave the cell. Now that means they have to get into the machinery that actually produces that translates and transcribes the genetic information and makes proteins at the end of the day, and then leads to a cell budding. All of that now is taken a little bit away from the outside in the inside of. And that, so there are two different protection mechanisms. Any other explanation why could it be an advantage to have a subclassification, to have groups underneath? So again, this is also enclosed by membrane. These are enclosed by membrane. The particles in here. Can you think about another reason why that could be an advantage?
Reproduction.
What? How?
Could be easier if the genome is concentrated
on a closer space. That's a very interesting idea. I never thought about that. That's a very good idea, because in fact you do what the image sort of suggests. There's a scale difference. Bacterial cells or prokaryotes are much smaller than eukaryotic cells, and maybe cell budding would actually get complicated if the entire DNA would sort of swim around in the cell. That's a very good point. Never thought about that. Point taken. I do not know anything about the biology of that, but yeah. That could be a reason. Any other idea? Yeah?
Maybe because different different compartments, we have different environments. For example, the backseat, some part is not very backseat.
Why would that be good? That is true. In fact, you state things that are absolutely right. But why could that help?
Make it more effective to specific.
Make what more? That you are completely on the right track here. Can Jinlong, can you go one step more? Make what more effective?
So in different environments, they can do specific jobs effectively.
Exactly. So in the mitochondria, I said everything that has to do with energy production happens, right? And there's a particular type of process, and you can optimize the entire process. So single steps in the process, but it can actually optimize the entire process to the local environment. Okay? Now, this is one important aspect. Another important aspect of this sort of same idea is not only because you're sort of making things fit to the particular local environment, but also because you actually, back to and now comes the phase where I'm trying to learn the names. Back to what Mahler sort of implicitly said, by putting the particular process into one environment, you can also focus them better. So you can have more efficient processes. More efficient processes, for instance, is that if you have a protein A that is used for some process in DNA processing, and the same enzyme happens to be used for something that relates to energy, if all swims around in the bacterium, you have to make sure that they will not confuse each other, right? That the one will not take away from the process of the other one. Well, here these processes are completely independent. And that's another advantage. So you will have the right protein in the right place at the right moment, more likely, if you're focusing on some processes. Again, part of what Mala said is also the size, or Mala's point is about the size too. So maybe you have to have this subdivision also because, in fact, in order to blow up to the bigger scale, otherwise things would get too complicated. But anyway, so we see a couple of reasons why these compartments help. And that again means that in the protein there is some signature how that protein actually gets into that compartment. For most of what we will do today and next Tuesday, we will sort of focus on the idea of a sort of three-state system. So in particular, we will sort of forget about the mitochondria. We'll just think about extracellular, cytoplasmic, so inside the cell, and nuclear, so anything that has to do with DNA. So the means by which we can do it is by which we can predict for a particular protein its subcellular localization, so where in the cell it ends up. First of all, is by homology. So by homology means I know another protein that is very sequence similar, for which I experimentally know in which localization that is. So the particular human protein and the particular, bless you, for the mouse protein I have some experimental results. So I say the human protein is also going to be nuclear. Something like that, right? Another one here is text similarity. So text similarity is if I have a bunch of descriptors that describe the function of this. So I said that in the nucleus, for instance, this specifically does everything that has to do with processing DNA, right? Now, if your protein has the description of DNA processing and all kinds of these words that relate to that, you assume that that protein is affected also in the nucleus. Okay, and we will get to that. Then there are the motifs. Now, motifs is essentially the idea of a zip code. So you have your protein, and I will come back to that in a little bit more detail later. So essentially, you put a zip code on a protein, and this zip code is the way in which the protein is essentially finding its right target. How do you believe? So this is an idea that goes back. The name is currently escaping me. It's on a slide, but I can't take it. So why? How could you imagine? So I'm giving you the idea of a zip code. So zip code essentially on a letter, right? In the letter, you know the postman is handling it, or the machinery that processes it. So if you had something such as a zip code on a protein, who would actually read it? Certainly not the postman, yeah?
I would assume that there is one protein which would be responsible for transporting other proteins.
Exactly. So it's another protein that essentially recognizes the zip code and says this protein is now going to be imported into the nucleus. And I'm going to show you in particular for the nucleus and for a variety of other compartments how this works. And then there is, of course, is the way of de novo prediction. So I could sort of look at the structure, and again, Jinlong said that different compartments have different pH values, different environments. So maybe the structure is compatible with a particular, if I look at the protein structure, I see the surface is such that it sort of fits to a particular environment, or not. Then I possibly can guess from that environment, or I can just look at the sequence. Since proteins are finding their right place, some without zip codes, we believe, finding the right place from just from their sequence, we should be able to do the same thing. So let's go back to homology-based inference. Again, the similarity between a protein of experimental annotation and a query is higher than some threshold, and then I annotate the annotation of E for Q. This will always work very well if you have very high levels of sequence identity, and will work at some point you will lose the signal So the twilight zone is the zone that essentially is defined as the one when I can no longer, without many mistakes, distinguish between making this decision right or not. And the midnight zone is essentially where the classification gets to the level of random. In order to see where the threshold T in this equation is, so that's the major unknown here, we need to essentially start with the set of proteins for which we know experimentally the subset localization, cluster it such that I have a sequence unique subset. And the clustering has to be true, because looking this set up, they could all be 100% sequence identical to each other, and I would never learn anything about T, right? So I need to get at a set here, a subset that is such that in that subset, no pair is similar to each other. Now, what does it mean, similar to each other? Well, one operational definition is that any pair in there is less than T. And we're in a circle, right? Why are we in a circle?
You start clustering to and create a subset to find T, and you need T in order to create a subset. So
how can you get out of that? We learned it in math from induction, essentially. So what you try is you use a T that you believe is way, way lower than the one that you will then at the end find. So you make this much more conservatively unique than maybe in the end you will find. And then at least you did not hurt yourself, and at least the T that you find is right. So everybody understands that statement? So again, let's go back to this T here. If I need the clustering to find out what the T is, and then I say I use the T for clustering, I have a circle, right? So one way of doing that is I choose this T from when can I do a homology-based inference for inferring that two proteins have a similar structure. Because I believe that the T for a similar structure will be much lower than the T for similar localization. Proteins with a similar sequence, with fairly different sequences, still have similar structures. But some proteins with fairly similar sequences already don't have similar localizations anymore. So I assume that the T, let me just physically write that out. The T for inferring that two proteins have a similar 3D structure, let's call it 3D, is substantially larger than the T that I will need for similar subset localization. If that is the case, so we have to revisit in the end when I find my T whether that was true. But if that turns out to be the case, then in my first clustering here, I will simply take the T3D. And I will do a conservative estimate. Okay? Everybody clear about that? Okay, so then I have to simply compare everyone in this set against everyone in this set, and simply see whether I sort of can infer subset localization. And that is one plot where I show the HSSP distance, number of residues aligned, percentage sequence identity. The blue line here is the so-called HSSP curve. That's an empiric fit into distinguishing between proteins above the line that have similar 3D structure and proteins below the line where we cannot say that they have similar structure. It's not that below the line they are proteins of different, but it's just we cannot say. The line is simply the twilight zone point of structure inference. Now, the difference or the distance to this line here is essentially you move the line by 5 percentage points up, get to that point, plus 10, plus 15, plus 20, minus 5, minus 10, and so forth. And that is the number you see here. So 0 is the blue line, plus 20 is 1, 2, 3, 4, 5 would be this line. Minus 20 would be this line here. Okay? And you see, so essentially blue is similar structure, and green is similar structure. Blue is similarity in subset localization. So it shows you that if you have a very, very high value here, so if you have very, very high sequence identity, mostly the pairs of proteins have a similar localization. It's not quite 100% here, so it deviates from 100% very early on, but it's very, very close to 100, and it begins to sort of fall at some point here. Now again, the T here, now it depends on you where you will put at the end of the day the T. You could say that, well, wherever it falls below 95, wherever it falls below 90, whatever you say, the blue here, the T for localization is much higher than the T for the green.
I have a point.
Yeah. Good luck. So the original assumption was right. So we couldn't do the clustering the way we did it because most likely a safe threshold is somewhere up here. Okay, now here is a sort of similar curve, but in this particular case for a different level of measuring sequence similarity. So here's the distance from this curve, and here it's a log E expectation value from the so-called psi-blast alignment method, where in fact the distinction is slightly worse. So that's why this works better. But anyway, so that gives you the T. Now, if you want to predict different aspects of function, will you take the same T? So now I establish a T for subcellular localization. In my next project, I want to predict that a protein is an enzyme. And I again ask, what is the T? Which T will I use? So in this particular case, the slide already has an answer to that point. For the enzyme, that's the red and orange. So we actually have to redo that for enzymes. And the downside, so the upside to this is that it's extremely powerful. The downside is you have to essentially, for every single aspect of function that you want to sort of predict, in principle you should redo the T. And you do see that whether it's, so these four curves here, they look different. So it's clearly, you cannot clearly not take exactly the same T. Okay, let's leave it there. Let's get to the issue of text analysis, what I meant there, and that is a project that was started by Rajesh Nair, who is now at the FDA. So Rajesh simply looked at this record and said, okay, now if in some cases we have all of this information, except for we don't have this column subcellular localization. So this is an entry from a database called Swissprot. Swissprot is an annotated version of a protein database. So in terms of numbers, UniProt is the database of all proteins. At this point has about 150 million. Swissprot is annotated. Annotated means that there is an expert, a molecular biologist, who reads papers and puts up a Swissprot record that summarizes whatever they find in that paper, or papers, for one particular protein. And therefore the number here is in the ballpark of 500K. So much, much, much smaller. And this is not because there's not a lot of work going into Swissprot. In fact, there are many people working on Swissprot. There are many annotators. And so this could be an extraction, but it's a controlled vocabulary. So it's not complete wild text, it's not free text, it's not quotes from the papers. It is looking for functional criteria, for functional descriptors that are already contained in the database. So they have a list of descriptors, and then annotators are encouraged to sort of use them. If people discover new functions they will add to the list, right? But in that sense it's a controlled vocabulary. So keywords repeat within the 500,000. That's one way of saying it. Okay? Now, the idea simply was use this information. So how could you do that? So let's begin from the first idea. The first one, let's call it as trivial, and we already talked about that. If you say DNA binding or chromatin regulator, experts would know that this is nuclear. Blood coagulation that is going outside the cell, so secreted, because the blood is outside of a cell. So anything that has to do with blood coagulation has to be outside. Now, the goal of this method that uses these keywords is to find something that is not trivial. So in that sense, trivial is if I have a particular keyword and is 100% linked to a particular subcellular localization. That's trivial, right? And at the same time, it's trivial because a biologist looks at it and says, this is clear, we know that. At the same time, somehow, discover what is trivial for some expert, but not for the machine. So these two things we want to somehow get out, right? And they're of course related. But how could you do it? So first we begin, of course, by extracting keywords for all of these classes of subcellular localization. And we see some counts. We have thousands of those, and we now sort of we have 15,000 proteins for which we have some keywords, for which we can in fact learn, because for these 15,000 we do have the subcellular localization. So 15,000 proteins with keywords and localization. How can I now put that into a system? How can I learn to predict if I remove this keyword here, if I removed completely subcellular localization, to predict it from all of the others? How can we do that? Thank you very much.
Thank you.
But I will not go back and do the same thing. Thanks, Mala. I'm just excusing myself from not redoing it. Anyway, so we go one slide back. So we create all the subvectors, all the possible subvectors by permutations shown here, and then we sort of compare each subvector through that Shannon information, P log P, with all the other vectors and find the one that is highest. So then we essentially do a K-mer of the subvectors, right? K-mer match of the subvectors. The database is in that direction, and the search direction is in that direction. Yeah?
But how does it work? Because if you permutate the vectors, then you basically permutate the keywords, right?
No, wait, sorry. You turn to 0. So this is the point. Okay. So what you have to understand, the background idea here is, or the background biology in this particular case is, that not all the keywords are always present. Mm-hmm. So yes, you build your data set with the 15,000 proteins for which you know the keywords, and some of those, in fact, most likely they are annotated better than the one you will come in with tomorrow, because they are in the database for longer. There are more people who have had time to add keywords. So the number of keywords you will see, or the density of keywords, is higher in those than for whatever will be added at any point of time in the future. Again, if you are sitting in the database for some time, the number of keywords accumulate. That is the idea. So you essentially probe the question of, are some keywords less important than others? So what if I don't have that keyword?
Okay. So you only turn to 0 and not back to 1.
Correct. I should have said that differently.
So it's not like a permutation?
No, I used the wrong word.
Okay.
You're right. And that is exactly what is misleading in what I said. By the way, here's another thing I suppressed for simplicity. You're not using all the keywords. You're using only the most informative ones. So that has to do with presence in the data set, and has to do with how many, so keywords that map to all subcellular localizations are all kicked out, for instance. And that immediately sort of kicks out some numbers and things like that. Now, the system gets some performance for using the best hit, okay? Now, you can ask, okay, it finds the best hit. What about the second best hit? Is there a meaning in the second best hit? How would you test that? So what I'm going to show you on the next slide is I'm going to show you something that is sort of relating precision with recall. On that slide it's called accuracy and coverage, but essentially is precision and recall. Precision and recall. And in this precision recall curve, you have a curve for the top hit. Now, if I asked you the question, what about the second hit, what would you do? It's very simple.
I would just create a confusion matrix where I have the first hit on the...
Very good idea.
And then the second there, and look at, okay, do they usually agree, or where do I have confusions?
That's a very, very good idea, and I'm not going to show you that on the next slide, but thank you very much. You should always begin there, Sophie. I'm not showing you the confusion matrix. It's a very good point. You should always look at the confusion matrix. So again, the confusion matrix, for those of you who are not familiar with that term, is, well, essentially if we had let's just begin with the 3 subcellular localizations that I talked about: nuclear, cytoplasmic, extracellular. So there's one in say, the observation, and there's one in the prediction. So if your prediction is right, then all cases end up on the diagonal. If your prediction is wrong, so you have predicted it as nuclear, but it is observed as cytoplasmic, say this is 2 and this is 4 cases or something like that, then the confusion matrix would say, okay, 50 times are correctly predicted, 6 are incorrectly predicted, 2 of those 6 mistakes are where I believe it's nuclear, but it's actually cytoplasmic, and 4 are where it's actually extracellular. Now this teaches you a lot, in particular also from machine learning devices, because often what you will see is that only one class is populated, so that you essentially learned only one of the many classes you try to predict. Now my question really is, specifically, I want to look at the second best. So I'm not only going to ask, is my first hit best, but I'm going to ask, is my correct observation in one of the first top 2? How would you do that? And again, I'm going to have a ROC curve. Well, now I sort of said it. You essentially plot the second hit here, the red one, right? Precision, recall. So, is the red better than the blue? Can just say something. The question is the red, so the red is the ROC curve, or ROC-like curve that you get for the precision recall curve, that you get for, is the correct hit in the top 2? Is the correct prediction in the top 2 hits? Is the correct prediction in the top hit? That's the blue. Now my question is the red better than the blue? yeah. So Tobias and Sophie have both venture to answer the question, but they have different answers. And on a binary decision, this is very easy to see. Because suddenly the difference gets extreme. Okay, let's argue for the yes.
What does coverage mean in this case?
Recall.
Recall.
Precision, recall. Right. So what you want to be, in order to be better, you have to be above. Red is above blue, and that's the yes part of the answer. The no part of the answer, don't you say it yourself?
I guess the ideal curve would be below the red one. So it's
above, or what do you mean?
No, below. Like the ideal ROC curve looks something like.
Oh, I don't have ROC here. I don't have numbers. So this is essentially precision versus recall. Random is not a line, not a straight line here.
Yeah, okay, I see.
I do not know what random is, so that depends on the number of states. If the number of states would be 5, and I can't remember how many states we have in this, I do believe it's more than 5. But if the number were 5, then the random is sort of 20% accuracy. So you see that accuracy in this particular case here ends at 80. So random would be somewhere.
Yeah. Below.
We are clearly above random. Yeah? Okay, so we still only have one answer. So we still only have Sophie's yes answer. The no just sank into collapsed. Yeah?
I would just like to know that the problem is that the top 2 hits, here we only measure what the correct answer is in one of those 2 predictions, but that doesn't actually help us for real predictions, because then we still don't know which one to pick.
That's true. But okay, so if you, again, these tools are typically to help experimentalists do their experiments. If the experimentalist told you, well, I can work on 2, as long as the right one is there, then it would be relevant to look at this, how good are you with the second best hit? So how many cases can you get? And you can say the experimentalist wants a sort of higher accuracy, at least I cover a third. 35% or something like that at a high accuracy. So I can get 86% accuracy, and I find 30% of the cases. So I'm not finding 100% or 35% of the cases. I'm not finding them all, but at least for some, I could give you two. I give you two, and there's a 1 in 3 that you actually find a good result or an interesting result. And maybe the experimentalist then says yes. When I say, okay, I give you I give you only one, but then 20% of those ones that I give you are wrong. Maybe at some point, well, whatever. There's a threshold, right? Clearly, the red curve is above the blue, so whatever the answer is, that may make a difference. It may not make a difference, but it may make a difference, okay? So there is a biological or an expertise or whatever. There's a scientific reason why you may want to look at the second hit. And you are right, it still doesn't give you the final prediction, but maybe that's good enough. My question is it better? Again, numerically higher. Red is above blue. But in order to make the point of better, I need to consider something else. And that goes back to the random story. So in order to really say that this second one is better, I need to say that it adds more than a random guess of the next one would add. Since I allow myself to pick 2 and not only 1, the random probability changes. So for the first hit, if I have 5 states, for the first hits, this is 1 in 5, right? For the second hit, it's already 2 in 5. That means this one has to get up by more than 20% to beat, in as much as it gets better than blue, to beat a random addition. And that is not the case in this particular case, right? So in that particular case, then you would argue this is actually not better. So the method is really only good at finding the top hit. It's not good at sort of estimating the probability of the hit or at going down further the list. Or you could almost replace it by random, right? And that is an extreme way of saying the method doesn't really help you. And you should look at, whenever your method puts out a probability, is that probability meaningful? And this is what I wanted to bring up here. So if you take 2 out of 5 or 1 out of 5, the background probability changes, and you have to take that into account when you answer the question whether one is better than the other. Okay. The method can sort of identify some tricky correlations that even experts did not quite clearly see that way, and we can use it. And now we get into the motif story, or the Zipcode idea. The Zipcode idea, there are 2 types of motifs, or zip codes if you want. So one is sequential, meaning that there is somewhere on the sequence a motif. I showed it with this flag in my drawing here, my doodling, that is recognized by another plotting. Okay? And then there is sort of a structural motif. The structural motif is such that only when you fold to a 3D structure, you get a motif of these 2 red regions. Together they form a motif, because they come close in space. Okay, it could be, for instance, this is very positive charges. So you put 2 regions together that both have 2 charged residues. Now, the 2 charged residues on their own is not enough, but if they fold together 4 charged, suddenly that gives a motif, and that is relevant for something. If you pull a little bit more, the screen is going off. Anyway, so
does this mean that you'd have to know the 3D structure to know the motif?
Exactly. And that in fact turns out to be the limiting step here, and ultimately for this particular topic that is the end of the story, because most of the methods that we present are sequence-based without having to know the 3D structure. So there are some of these motifs known, but they are very difficult to identify because you have to know the 3D structure, and that makes it more complicated. So the only kind of thing where this actually works is to identify binding sites. But for targeting motifs, we know some examples. Okay, so that's one reality. The other reality is that there is a trafficking in the cell. So there's a particular, let's call it pathway of trafficking. Proteins move from the inner cell, the cytosol, in through to the nucleus, and that is sort of a gated, there are gates there, a gated exchange, that's why it's red, it's sort of a different path. And then they are going to the mitochondria, they're staying there. They're going to peroxisomes, they're staying there. They're going to plastides and staying there. And then there is another path, this one here, that goes to the endoplasmic reticulum, then onto the Golgi, and then ultimately to the cell surface. So there's a bunch of places in the cell where a protein could end up along what is considered the secretory pathway. Many of these proteins that begin here, they go through all of these compartments here, and then they ultimately end up outside the cell. You may call that a very regulated way to export. It is. And I'm not entirely sure about, I said most, I cannot quite remember the numbers, but the fraction of proteins that stays here or here is relatively small compared to the ones that go outside. Okay, now this particular pathway, the secretory pathway, is in fact characterized by something that is called a signal peptide. And the word is not so important, the idea is. So that really is the idea of a zip code. And it's a zip code that is at the begin. So what we know about it, they are typically the first 15 to 30 residues. At the N-terminus means the beginning. So biologists refer to the protein sequence as N-terminal and C-terminal. This is simply because of the chemistry, the nitrate or the carbon. Anyway, the N-terminus is the first residue in the beginning of 15 to 30 residues. Often make up the signal peptide. Many of these signal peptides are cleaved. Cleaved means cuts. So essentially that is a zip code that is relevant only for getting outside, for instance. If your destination is to go outside of the cell, the moment you essentially, just before you get outside, it's cut off. And the native protein then will be a protein that has the same sequence without this zip code. And that will then go outside. Okay, they exist in all three kingdoms, so also bacteria, and they have a fairly simple, I refer to it as an architecture or a system. And what I mean by that simply is, so first of all, there are 4 different regions that you can distinguish, called N, H, C, and cleavage site. So these are regions, and the cleavage site essentially is a particular residue, one or several. The N region often has charges, positive charges. The H region is hydrophobic, typically is more than 6 residues, has a lot of hydrophobic residues, can be 10, 15, 20 hydrophobic residues long. The C region is polar and uncharged. So we define these regions as prevalence of a particular type of residues, and they were defined by some expert who had looked at it, so essentially collected all of these sequences, and so okay, let's go back one step. So you have signal peptides. Signal peptides are, I'm going to tell you, are terminal, N-terminus, beginning of a protein. How can you discover them? So if the person who I refer to here is called Henrik Nielsen, and when Henrik Nielsen started this about 20-something years ago, there was no database available. So Henrik could not have gone to just find a database. When we started in this project, we had a database, namely the one that Henrik had built. And then, well, how would you go about sort of identifying the motive of a signal peptide? Somebody gives you 1,000 of these signal peptides, and how would you find the motive? You could do machine learning. Let's not assume that you can. Let's just assume you really want to find motifs. Motives. So local rules or something like that.
I guess you could assume, or at least suspect, that they are always at the end or at the beginning.
Yeah.
Then cut, like, I don't know,
30 residues, yeah.
From the end or the beginning. Then just, I mean, if it's a really simple method, without computer, you can just list them and see, like, beneath each other and see if there are patterns which are obvious to you.
But how would you see these patterns? That's exactly the question.
Like,
Motif beyond something that is described this way. So it is essentially what you said is sort of an expert looking at it and seeing some biophysical features, but trying to go beyond that, at least in our hands, didn't quite work. And so one way in which you can do it, and ultimately that's what you said, you chop them apart and you see, for instance, are there particular letters that are particular amino acids that are used more often, and you see some motifs, you see some things. But essentially this one here you only see in that clarity for the cleavage site. So the position where another protein will dock and cut this zip code off. And that you can actually do through these profiles, through the motifs. But everything else is not much more than that. What you, however, can do with that, now you can build machine learning devices, neural networks, and later they used hidden Markov models, and then they went back to neural networks. And sort of this is a very, very successful method. And we don't let them in. So there are many, many, many other methods that do it. But ultimately, the initial work here really is still the state of the art. There are other types of signal peptides. There are other types of signals that are like that. So the one I talked about is secreted. There's something that's called a transit peptide. There is a compartment in plants called chloroplast. So it's a sub-compartment inside of the eukaryotic cell that exists only in plants. As for chlorophyll, so it's plant-specific process or job. And that also, the regulation, so the proteins that go into the chloroplast are also regulated through a motif like that is N-terminal, so it's at the beginning of the protein, and looks similar to what the signal peptide looks. Just because this is biology, there's a different name for it. And then there's something in the mitochondria that is also like that. And again, it's called targeting peptide in this particular case. So signal peptide, transit peptide, targeting peptide. Ultimately, they are all the same. They're not quite the same in the sense they are not all exactly the same length. The cleavage site is they're not cleaved. The particular composition is slightly different, but essentially they are the same, and they are all such that you can machine learn them. Okay? There's something in data, so you just collect enough of these motifs, and then you can machine learn this and predict it for new motifs. And now we get to the nuclear localization signal, and yet another word for the same idea. The nuclear localization is just like the signal peptide, something that is extremely sequence-based. So it's not 3D, it's visible on the sequence. And the idea is you bind, so NLS, nuclear localization signal, there's a protein called importin that binds to the nuclear localization signal, transports your protein into this nucleus. There's a double layer of membranes here, and there's a gated traffic, so there really are channels, gates here to get into the nucleus. Okay, and that is the way you do it. And so nuclear localization signals are very extreme, I was about to say, in the sense that you take a short peptide, a nuclear localization signal, you put it onto a piece of gold, and the gold is put into the nucleus. So anything that has this zip code, importin will grab and put into the nucleus, no matter what it is. Okay? So now you want to sort of build your data set of nuclear localization signals. And so there was a database for motifs out there when we started and wanted to do that, called ProCite. And ProCite had exactly one of these. There's a so-called bipartite motif, but bipartite means there's a little bit here and a little bit there, so there's a little separation of these two parts in sequence. And so that was matching in 96 proteins at the time. They refer to 31 families. So proteins can be clustered into families. So this is sort of the diversity. The one motif matched in 31 completely different families. completely, at least 31 completely different proteins. The accuracy was 90%, so precision is 90%, so meaning that 90% of the stuff it matched was really nuclear, experimentally, and had a 3% coverage or recall, meaning of all the proteins that were known to be nuclear at the time, 3% were matched by this particular motif. Okay. So then we went out and simply read the literature and collected motifs. And these are some of those. Red, you see a positive charge. So the red marks a positive charge. You immediately see that there is a lot of red, but you also immediately see that there's a lot of size difference. So we are no longer at the point where we just cut the first 30 or something like that, or in this particular case shorter. Some of these here are extremely or relatively short, some of them are longer. Here what you hide is any 18 in between, so that's a bipartite motif. So you have something that is constrained and then anything 18 for 18 times and then something that is constrained again. And then further, now you see that the diversity gets even larger. So this is the kind of thing where you can clearly not machine learn. This is too much for at least. So I did not give you the full list, but it's not far from the full list that I gave you that we collected. So this is too complex and too small. So if you had millions of these, maybe you again could sort of machine learn subclasses. But on the list that I showed you, you clearly cannot machine learn, given the diversity that we have. So then what we did is we did something. Now one issue here is, this one is one issue, suggests one issue. So you have a fairly short motif, and you have one positive, so what is a theme is that there is a lot of positive charges. This one has only one. It's very short and has only one. And that kind of thing makes me wonder, and here we have a longer one, but also only one positive charge. Makes me wonder how good the experimental values are. How could you find out? So again, we get these from reading a paper in which the authors essentially said that I identified this part as the reason why that particular protein is nuclear. It's really very simple.
Look at the paper, see if they make a mistake.
Well, typically those papers are carefully describing the experimental background. So what kind of mistakes? So that the mistake is wrong and that it's not a nuclear protein. That could be. But there's something else we have to consider here. So these people, in fact, do not consider who do the experiment. Their idea is, so what they want to know, these experimental people, is I want to show that my protein is in the nucleus, and there's this particular region that is the reason why that protein is in the nucleus. That's the goal of the experiment, right? Show that protein is nuclear, and B, that is the region why it is in the nucleus. That's the zip code, so to speak. Now I get this as a nuclear localization signal. What do I have to do with it? Well, I have to sort of archive it, put it into a database. I have it on, that is my database here, the whiteboard. Yeah.
Check if other...
Sorry, I was too slow. Sophie was too busy looking at you.
My idea would have been to look at whether this same motif also occurs in proteins which are not in the nucleus.
Exactly. So that particular experimental group is not really looking at, okay, that's my motif, how is that looking at different proteins? They only care, in my protein, if I mutate that motif, then the protein is no longer nuclear, hence this is the reason why. That's the way the rationale somehow works. But the question that you as a person with a computer have, or the task, is to figure out how generic is that? Do I find that same motif in other proteins? And I do some, what we coined in silico mutagenesis. So what we do is, we run this particular motif and see whether it hits only in nuclear proteins. If it hits in any protein that is not nuclear, so we want to crank out from this device here, at the end we want to have a set of motifs that are really absolutely nuclear only. That means if I match it to any protein that is experimentally known not to be nuclear, out. Now, motifs that are only matched in one particular family of nuclear proteins, they are good, but not good enough because we want something that is sort of generic. Okay? So now we're not quite, when I said out, we're not quite throwing it out. What we're going to do is, well, if it matches in a protein that is not nuclear, maybe this was right here. That is sort of the region, but it matches something that is not nuclear. So what should I do? If that contains some grain of truth, but it's not the whole truth, what is missing? What do I have to do?
Maybe it's only part of the motif.
Exactly. Make it a little bit longer, right? So look at that particular protein, look left and right. Is there something else that could be a motif? And that ideally would be something, if I look at an alignment, that would be sort of conserved. I find that in many because it's important. Because the nuclear localization signal is something important for transport, so you would expect it to be in many families. So if it matched in non-nuclear proteins, first we try to make it longer. Okay? And if that failed, then we threw it out. And then making it longer, we made it so long that it never matched anything here. And we tried to match at least two families. So matching these two families is the opposite problem. Maybe this one is too long. And then we have to sort of shorten it. Again, in the direction of conservation in the alignment. And then we sort of go through this iteration several times, essentially manually, on a computer, and get a database of motifs that at that particular point in time contained 200 motifs that matched in 186 rather than 31 families, and were by definition or by construction, the precision was 100%. And they mapped to 40% of all the more than 40% of all the non-nuclear proteins at the time. So over time, this sort of was 18 years ago, and we have just published an updated version of that. And these estimates were no longer true at all. So with the growth of the database, it had begun to map in many, many other proteins. And so we had to refine it. Now, one of the nice things about nuclear localization signals is, so what I show you here is a 3D structure of one of the motifs that we found. So that's the motif. Gray is the protein. Can anybody recognize what this thing is? No? DNA. So, any suggestion from this image here? So the gray, so this is on part of the gray, right? This is the nuclear localization signal that is on the gray protein. That is the DNA that the protein binds. So, what is this? This is the nuclear localization signal, but can you say something else about it? Technically, again, this is one of these NLS motifs that we have in our database. Okay, one of these 214. It's not obvious? No, it's obviously not obvious. Is it? No, I mean this is not obvious, right? Okay. Where is it in the protein?
At the end of
well, yeah, and it on this image looks end. It's not quite true, but
yeah.
But more importantly, what is clearly the case is it is binding to the DNA. Right? It's completely wrapping into the DNA. In fact, if you ask, giving this image, what are the binding sites of that protein binding DNA? It could be that there is one here, or here, that this long helix here makes a contact, right? We cannot see that from this image because it could be far away. It could be just a projection issue. But projection issue or not, this one here clearly sort of is fitting into the groove. right? That fits snugly into the DNA. So why would the binding site be there? And that got us to another one. In step 1, importin binds to the nuclear localization signal, brings it into the nucleus. Lo and behold, in the nucleus you have the DNA. Many proteins that are in the nucleus bind DNA. So the binding of the DNA, since in the second step the NLS goes off, the importin goes off, the NLS, so meaning that the NLS is sitting there again. And now it has to bind DNA. Why not use the same motif? That thing now binds DNA with the same site, right? So it's the site. Now we have a zip code that is recognized by importin for a protein that has to go into the nucleus. That protein then, importin goes off, that zip code becomes available and binds DNA. So use the same thing. The same motif with which you bind DNA is the one that is recognized to bring it into the nucleus. Makes a lot of sense. Because then the importin is essentially specialized on recognizing something that proteins that bind DNA have to have anyway. Right? And in fact, the cycle goes off. In some cases, there is a sort of export signal. So there's an exportin, there's an importin and an exportin, and the exportin recognizes again after it has bound DNA, same site again, and they export it again. This is not true for all nuclear localization signals, but some of those clearly are like that. So you got, with this Motif-based database, you also got for free a DNA prediction method. Okay, so then we get into this issue of de novo prediction of global signals. And now we get to the name Günter Blobel, who I could not find. So Günter Blobel got the Nobel Prize for sort of this idea and working out how proteins are transported through the cell, and this idea of the zip code that comes from Günter Blobel. That's the mentioning in the Nobel Prize of 1999. Proteins have intrinsic signals that govern their transport and localization in the cell. Now, fact is, so Günther published this in the original papers in the 60s here that I should quote, that is awarded in this Nobel Prize, so it's a while back. Nevertheless, and he got a Nobel Prize, so we all believe in this, nevertheless, the signals are not known for most proteins. Well, this could be because we haven't studied enough, okay? This is one issue. One possible issue is that there are new types of motifs, and that is the motif where we need really to see the 3D structure. Since for most proteins we still don't have 3D structure, that's the conformational. It will be difficult to find those motifs, right? Or there could be other mechanisms for the import. So can you think about a mechanism that would explain why for many proteins that are, for instance, going into the nucleus, we still don't know the motifs? And what kind of simple thing could you think about? So I want to understand, I find many proteins in the nucleus that don't have an NLS that I know. People have studied for many years these nuclear localization signals, so I wonder why they actually don't know it. Again, maybe some remain to be discovered, but if it is more than 50% after such a long time, you wonder, are there other ways? Are there other reasons?
I remember the bacterium which can shoot DNA through membranes without actually having such kind of a motive.
Yes, actually, those proteins are secreted, so they have a signal peptide. But anyway, but they don't have a nuclear localization signal, and it's true, they are completely different mechanisms. And they exist, and they are, and in fact, bacteria shoot through 2 levels of walls. So this shooting is immensely strong. So they're amazing. So this warfare, the bacterial warfare that is going on, is really amazing. If you laugh about my term of warfare, so if you have a bacterium that sort of swims by another bacterium, and this bacterium here builds up a cannon that is essentially as wide as the bacterium, all right? And it takes a long time. It builds it anew every single time it shoots. So it's a lot of energy and a lot of time involved. Then it shoots here. Now, that bacterium here is shooting back, okay? Where do you believe it shoots back from? It uses the same point. The same hole shoots back. Through that hole it shoots back. Now, if you sort of deprive them of their ammunition, what's going to happen is, so if you deprive, this is A, the aggressor, and this is the defender here. Now you remove, defender shoots back at A. And you remove all the bullets. So bullets are proteins here. That is also for DNA, but in most cases for this kind of warfare, they really are shooting proteins. So you remove all the proteins that it could shoot at A. What do you believe is going to happen? What is D going to do? Just give up? No. It's going to...
It's the same. It uses basically the missile of A.
Exactly. It shoots the bullets from A and shoots back at A with the proteins that A shot at it. So really, in that sense, you smiled, but this is really warfare. Anyway, but I meant something simpler. So the simpler thing would be, what I would mean by something simpler, is, so you have a protein with a cargo, and I actually, so I did that wrong here. My drawing is wrong. So you have the shuttle protein that recognizes the, I don't know why I did the drawing wrong and why I never learned that I did it wrong. So the green one should be here. So what I mean is that this one, no, that's right. The image is right. So that protein has a zip code. That protein binds to another protein. And that protein binding has nothing to do with the zip code. Since that zip code here is recognized by the shuttle protein, this one will be imported to the nucleus too, the green one, simply because it's stuck. So this would be a way to import something into the nucleus without a zip code. And if every protein had one cargo, then sort of you would only see 50% of the zip codes. Because 50% would sort of transport something else. And that may be, in fact, the major issue. And the question would become, how can we estimate the numbers? So how likely is it so that we have a second cargo? How could we do that? Could we estimate how many secondary cargo there could be? And this is something we can think about. But while we consider that, and we want to predict, yes?
Does it mean for the emissions you showed before that they can only enter if they're both binded?
Yes.
So they cannot enter if they're separate?
That is correct. That's
the binding is the reason.
Yes.
But we can only find the zip code in the first.
Yes. So this would explain the observation. So we have worked for many, many years, so we still have only 30% of the zip codes or something like that. Maybe because of that. And then what you would have to do is sort of look at how many nuclear proteins that have zip codes bind to other proteins, and then how many of these proteins they bind to in fact are nuclear. And this way you could sort of estimate some of the probability that this model could be right, and it makes sense. Unfortunately, there are too many unknowns for us to really give estimates that tell us, okay, every protein needs to have at least one cargo, or so many nuclear localization signals we expect. We're still not through with this, thinking this through. While we're not through with thinking this through and working this through, we should really begin with the novo predictions. And that's what I'm going to talk about on Tuesday. Thank you for your attention.